Hello, my name is Cameron Calder. I'm from the NetPhys lab directed by Dr. Javaria Hashmi, and I'm excited to share with you today my work on how chronic pain uncouples functional brain network segregation from cognitive performance in aging. Um, I have no relationships with financial sponsors to disclose. This project was supported by the CAHR, the NSHA, and the Brain Repair Center, but I have no conflicts of interest to declare, and no bias mitigation plan was required. So, chronic pain and aging have a complex relationship, and one facet of this is the impact of chronic pain on cognitive aging. On one hand, the presence of chronic pain has been associated with accelerated cognitive decline and a higher risk of dementia. On the other, poor cognition can be a risk factor for chronic pain and is related to worse pain-related outcomes. One strong explanation for these relationships is the accelerated brain aging framework. Chronic pain patients show structural patterns of brain aging beyond what would be expected for their actual age. These age-like structural features have in turn been associated with the degree of cognitive decline in older adults with chronic pain. Will these structural findings provide a good starting point? Understanding how chronic pain affects the aging trajectory of brain function will help us better understand and characterize cognitive dysfunction in this group. Well, measuring brain function can mean many different things. In my research, I focused on the concept of functional connectivity. Structural connectivity in the brain is enacted through white matter tracts, while functional connectivity measures the coordinated activity between two brain regions. A functional network has high functional connectivity between its regions, and these networks are theoretically important in coordinating brain activity in support of specialized cognitive functions. What system segregation measures is the organization of brain networks, specifically to the degree to which the functional connectivity of a network's regions are segregated within that network. This measure is one of the most reliable biomarkers of functional brain aging. Consistently, segregation declines with age, and lower or declining segregation have been associated with lower or declines in cognitive performance and increased dementia risk. Thus, this measure is an unexplored but potentially valuable functional biomarker for understanding the abnormal cognitive aging in chronic pain. So my first objective was to just reproduce the previous findings that chronic pain accelerates cognitive aging, or more generally, that chronic pain moderates the effect of age on cognition. Then I wanted to investigate the role of system segregation in this process, and I started here with the healthy control model, where system segregation decreases with age, and since higher segregation is associated with better cognitive performance, system segregation in this model partially explains the cognitive decline with age. So chronic pain could alter this in two ways. Firstly, if system segregation declines faster with age, so would cognition. And secondly, chronic pain could alter the relationship between segregation and cognition. If chronic pain patients were, for example, more sensitive to differences in system segregation, this would explain a greater impact of age on cognition. So for my methods, we had 64 healthy controls and 157 total chronic pain patients. Our primary cognitive measure was working memory, which was assessed through the NBAC task. And then system segregation was derived from resting state functional MRI data for each participant. And I can give, I guess, yeah, I can give more details on that at one point. And for our statistics, group comparisons were done using the covariate ANOVAs, and age and head motion were included as covariates when relevant. So at baseline, there were no group differences in working memory and system segregation between healthy controls and chronic pain patients, emphasizing that the effect of chronic pain on these variables might not be direct, but how these variables change with the aging process. Here we report a significant conditional effect of age on NBAC accuracy, and this was only in the chronic pain group, but this was not a true moderation effect. We can't say here that chronic pain was accelerating cognitive decline with age because the interaction term was not significant, but still this is a indication of accelerated cognitive decline that we might better detect with a larger, more balanced sample size. Then we notice that the kind of opposite to what we were expecting is that the chronic pain group's decline in system segregation with age is reduced. It's flattened. And this was a significant moderation. So the difference in slopes was significant enough that there seems to be a reduced decline in segregation, system segregation with age. Um, and then similarly, we see kind of this reversed effect where in healthy controls, we see the standard finding that with higher system segregation, you get better working memory. But in chronic pain patients, this association was non-significant and almost seemed to be reversed. And again, here, the moderation effect of this was significant. Um, so what's going on? What can we conclude from all of this? So really the key takeaway is that instead of just exacerbated functional network decline, kind of which we would assume to be coupled with the anatomical decline, structural decline mentioned earlier, chronic pain seems to disrupt functional brain aging. And so given the theoretically important role of system segregation in the aging process of healthy controls, these abnormalities could potentially be impactful and could be used to identify subtypes of cognitive impact in chronic pain. Um, Going a little deeper, according to the compensation hypothesis, declining segregation with healthy aging provides a necessary response to the anatomical aging process. And if the flattened age segregation relationship in chronic pain reflects a failure of compensation, this would indicate an especially severe disruption when considering the structural abnormalities of chronic pain and would explain why the segregation cognition relationship might appear reversed. And then also, when considering the role of system segregation as a cognitive biomarker and a biomarker of dementia risk, especially, we should really start considering chronic pain as a factor in these analyses and considering whether the presence of chronic pain might change the meaning of system segregation as this biomarker. Thank you to the lab and my supervisor, Dr. Javaria Hashmi, and thank you in total. And then there's some extra stuff in case, but nobody has questions. I guess this is a question thing.